So let me first begin by thanking the organizers for this opportunity to speak and also for hosting this wonderful meeting for all this almost more than a decade now. And so I'm going to talk about a problem where I'll discuss theoretical analysis of cargo transport by catch-bonded motors in optical trapping assays. And this work has been done in collaboration with Naren Sundararajan, who was an MSc student in Pune University, Shaugato Guho, who was a PhD student at IIT Bombay, and Mithun Mitro at IIT Bombay. So let's just begin with what is catch bonding. It just refers to the phenomenon of increased lifetime of a bond on increasing load force. So this is, so as you increase the force, the bond lifetime increases, which is called catch bond. In contrast, in context of ligand protein binding, you have the other kind of bond, which is the slip bond, that is with increase of force, the bond lifetime decreases. And in context of biological systems, and in particular for dynein motor proteins inside the cell, this catch bonding behavior is observed. What you see here is that I plot the dissociation rate as a function of load force, constant load force, and you see that there is a regime. Initially it behaves like a slip bond, that is the dissociation rate increases as a function of force, and then there is a regime where it actually falls off as a function of force. There are of course other examples of this catch bond behavior that is observed in context of biological systems. So how do we model this catch bonding behavior in dynein and how it can be understood? So we had in fact a couple of years back proposed this TFBD model. So the main ingredient of this model is that here is a schematic of a microtubule filament, and here is this cargo, and when this dynein binds to this filament, if there is a load force which is applied, then there is this contraction in this AAA domain, which essentially leads to stiffening of the this stalk which attaches to the microtubule, and it manifests as that means the catch bond basically gets activated at some typical force scale, which we call FM. And so this model is this bond deformation energy that the catch bond activates. And so the parameters which characterize the catch bond is of course the threshold force at which it gets activated FM, and F0 and alpha are kind of reflect the strength of this catch bond. And to by fitting, we can reasonably reproduce the single dynein unbinding characteristics from the microtubule. And so this is the plot. Now the question that we are posing is that given that this kind of catch bond nature is observed in dynein, how does it manifest in context of different intracellular processes? Like for instance, what we have seen is that it can actually lead to very interesting behavior. So for instance, if you're thinking of a transport of a cargo by this kind of catch bonded motors in context of unidirectional transport on a microtubule filament, then you can have this anomalous behavior where as a function of increasing load force, constant load force, for certain regime the velocity can actually increase even when there is an opposing load force. Similarly, in context of bidirectional transport, in fact it can lead to this kind of very non-trivial behavior. And indeed, some of this model predictions that we had done, theoretical analysis, is a plausible way of resolving one of these long-standing paradoxes which goes by the name of paradox of codependence. And these are in context of intracellular transport. One can also see the effect of catch bond manifest itself for the stability of this kind of motor filament complexes, where it can lead to this kind of oscillations which are mediated by catch bond. But essentially all these studies were done for a constant force. So there was a constant load force, and what are the manifestations of the catch bond in context of these intracellular processes. But here I want to focus on this question of catch bond in context of transport characteristics in an optical trapping assays. These optical trapping assays have provided an useful means of actually finding out the single motor characteristics at the level of resolution of single molecules. And in that sense, these have proven to be very useful in studying the different, how the motility characteristics change as a function of changing the different motor characteristics. So this is sort of the schematic of this thing, and our aim is to model this thing and find out the different transport characteristics as a function of the catch bond. And the measures that we take into account are basically, I would be looking at the distribution functions of the first passage time and detachment force. How do I define first passage time? So imagine you have this, so initially, so maybe I should go to the setup. So this is the typical setup. So just imagine initially these motors can stochastically attach to the microtubule filament at 0 position, and then they can stochastically detach and attach, and in the meantime they can start walking. And then you can ask the question, what is the distribution of the times when it again detaches? You can also ask questions about the distribution of the typical detachment forces at which it detaches from this setup. Now, in general, this is the 2D schematic. So how you go about we implement is that you have to impose the force balance condition, and of course there are these additional assumptions that we invoke, that the motor velocity has a linear relation with force. And finally, the equivalent 1D schematic, which is of course an approximation, you can actually analytically find a way of calculating the entire distribution, the first passage distribution, and we compare it with stochastic simulation. So first of all, I would like to point out that whether I approximate it with 1D, at least for the typical forces and the motor parameters, there is very insignificant difference between these two analyses, and that's why you can see that this comparison is almost very close to each other. The interesting thing that we observe is that there is a non-monotonic behavior of the first passage time as a function of the velocity of the cargo in this kind of setting. And a subsequent analysis, so first we go about analyzing what are the transport characteristics for a cargo that is being carried by this single motor, which can bind and unbind in a force dependent manner. Next, we actually go into, and just to point out, so this behavior is in contrast. So if catch bonding was not there, then you would not have observed this kind of non-monotonic behavior of the first passage time. Then we actually try to compare it with the experiments which were done with this human DNA in this kind of optical trap setting, where this optical trap setting, you can think of it as a harmonic potential. And what I show here is that, and these are done for different trap stiffnesses. And what is seen here is that if I incorporate the catch bonding, then it is able to reproduce the experimental characteristics much better. It also tells you or hints of this possibility that, if you want to observe this catch bonded effect, the force scale at which catch bond sets in need not be same as the stall force of the motor. That means the force at which the motor stops. And this kind of effects of this catch bond we also studied in context of multiple motors by stochastic simulations. And essentially the main point is that this catch bonding can, first of all, of course, it leads to higher first passage time, which is not surprising, but it also leads to this kind of non-monotonic behavior of the first passage time as a function of motor velocity. And similar non-monotonic behavior is also observed as a function. So as a function of stall force, we also again observe the same kind of behavior. So in summary, what I have proposed here is that this catch bonding actually provides a means for regulating transport, and it manifests itself in context of these different transport characteristics in optical trap assays. And we observe this kind of non-monotonic behavior arising purely due to this catch bond, and it seems to match well with the experiments. And it also provides a means of estimating this catch bond force scale. So yeah, I leave it with that. I mean, this is something we were planning to, or in the process of actually doing in future. So I'll stop here and would be happy to take your questions. Thank you for your attention. Yes.
May I ask? So this, I didn't understand very well, but this catch bond, what you're saying has the dissociation law is different from the usual Bell's law. Is that the difference?
No, so usually for meaning catch bonding, it's just that if I apply a load force, in general for slip bond, the unbinding rate will exponentially increase with force. That's an example of slip bond. For catch bond, it's just the reverse.
So are you just applying changing the law and seeing what difference is there? Is that, or is it coming
I
from
am
some
incorporating the fact that at the individual level, this dynein binding, unbinding characteristics from the microtubule filament have this catch bonding, which we have quantified because of through comparison. And then what happens even at the level of this single dynein, what are the characteristics, and also multiple dynein attaching and detaching from a carbon.
So this, how prevalent are these catch bonds? For example, if I look at collagen or something, and look at cross-links, do they form catch bonds, or how do they behave?
No, so I don't know about the specific collagen system, but typically, like for instance, in context of this P-selectin binding to endothelial cells, they again exhibit catch bonding. But the conventional wisdom was that catch bonding essentially increases the sort of the affinity to remain attached even under conditions of tensile forces. What here the perspective is that for in context of intracellular processes, they have a role which goes beyond just improving the adhesion properties. They in fact play a role in even regulating the transport. So that was. Yes.
Okay. Another question. How sensitive these predictions will be to the trap stiffness? Suppose you do your calculations with different trap stiffness.
Yes.
How sensitive they will be?
Yes. So this is in fact for the low trap stiffness. In fact, what we see is when the trap stiffness is low, then whether it is catch bonded or not catch bonded doesn't seem to matter much. And in fact, you can see that. And on the other hand, at least for the single motor case. Whereas in this case, you can see, I mean, here I have given a comparison of the theory where I have taken the bond to be just like slip bond and the catch bond and compared.
No, I'm talking about the trap stiffness itself. You are considering, like, you can work with different amounts of trap
Yes,
stiffness.
so these
right?
two plots are, this is for a trap stiffness of, I think, 0.01 piconewton
Okay,
per
so
nanometer.
you have done
And the other one where you see this comparison where it agrees better, this is with the catch bonded case, that is for a higher trap
All right.
stiffness.
Okay. Thanks.
If there is no any other question, so let us thank the speaker again.
Thank you.